1665, a man named Robert Hooke sliced a piece of cork, put it under a crude microscope, and saw something no human had ever seen before: tiny, empty, box like compartments arranged in neat rows. They reminded him of the small rooms monks lived in, called celluli, so he named them cells. But here's the thing: what Hooke actually saw were dead cell walls. The living contents had long since dried up and vanished. He discovered the cell without ever seeing one alive. It took another scientist, Anton van Leeuwenhoek, to observe the first living cells. He improved microscope lenses until he could see bacteria swimming in pond water, protozoa crawling, even red blood cells flowing. He called them animalcules, tiny animals. And then, in 1831, Robert Brown looked inside orchid root cells and spotted something sitting in the center, a dense, round structure he called the nucleus. 3 discoveries, 3 different men, each peeling back another layer of a secret that had been hiding inside every living thing on the planet. These discoveries built toward one of the most important ideas in all of biology, the cell theory. In 1838, a German botanist named Matthias Schleiden studied plants and concluded that every plant is made of cells. 1 year later, in 1839, a German zoologist named Theodor Schwann studied animals and reached the same conclusion: every animal is made of cells too. Schwann also described a thin outer layer around animal cells, what we now call the plasma membrane. Together, Schleiden and Schwann stated that all living organisms are made of cells and that the cell is the basic unit of life. But their theory had a gap: they couldn't explain where new cells come from. That final piece came in 1855 from Rudolf Virchow, who declared omnis cellula e cellula, all cells arise from pre-existing cells. No spontaneous generation, no cells popping into existence from nothing. Every cell is born from a cell that came before it, like a chain of life stretching back billions of years. So the modern cell theory stands on 2 pillars. All living organisms are composed of cells and products of cells, and all cells arise from pre-existing cells. That's the idea that changed biology forever: the realization that every living thing, from the simplest bacterium to the largest whale, is built from these tiny units. But not all cells are built the same way, and understanding how they differ is where cell biology truly begins. Every cell on Earth falls into one of 2 categories, and this single division is the most fundamental in all of biology. Prokaryotic means before the nucleus. These cells have no membrane-bound nucleus. Their DNA just floats in a region called the nucleoid, naked and circular, without histone proteins wrapping it up. Think of a prokaryotic cell as a studio apartment, everything in one open room. No walls dividing the kitchen from the bedroom. They're small, typically 1 to 10 micrometers across, and they lack all the fancy membrane-bound organelles: no mitochondria, no endoplasmic reticulum, no Golgi apparatus. Their ribosomes are 70 smaller than eukaryotic ones. Examples include bacteria, cyanobacteria, and mycoplasma. And mycoplasma deserves special attention at just 0.3 micrometers. It's the smallest known cell capable of independent life. And it's the only bacterium that lacks a cell wall entirely. Eukaryotic means true nucleus. These cells have a well-defined nucleus wrapped in a double-layered nuclear envelope, with DNA organized into linear chromosomes wound around histone proteins. Think of a eukaryotic cell as a house with rooms. The nucleus is the study. The mitochondria are the power generators. The endoplasmic reticulum is the factory floor. And the Golgi apparatus is the packaging and shipping department. They're larger, 10 to 100 micrometers, and their ribosomes are 80s. All plants, animals, fungi, and protists are eukaryotic. But here's a detail that trips students up. The mitochondria and chloroplasts inside eukaryotic cells have their own 70s ribosomes, a leftover from their ancient prokaryotic ancestors. Within eukaryotes, plant and animal cells differ in 3 key ways. Plant cells have a rigid cell wall made of cellulose, chloroplasts for photosynthesis, and a large central vacuole that fills most of the cell, like a water balloon taking up an entire room. Animal cells skip all 3: no wall, no chloroplasts, no giant vacuole, but they have centrioles for cell division that plant cells lack. Finally, cells come in a staggering range of sizes and shapes, and in every case, the shape follows the function. The smallest cells, mycoplasma, are just 0.3 micrometers. The largest single cell on Earth is the ostrich egg, visible to the naked eye and several centimeters across. The longest cells are neurons, which can stretch over a meter from the spinal cord to the tip of the toe. Imagine a single cell as long as a guitar. Red blood cells are biconcave discs, shaped like deflated balls to maximize surface area for carrying oxygen. White blood cells are amoeboid, blobby and flexible, so they can squeeze between tissue gaps to hunt pathogens. Nerve cells are branched like tree roots to collect and transmit signals. Smooth muscle cells are spindle-shaped, tapered at both ends for efficient contraction. And that's the foundation of all cell biology. Every living thing on this planet, from a bacterium 0.3 micrometers wide to a blue whale 30 meters long, is built from cells. Some simple, some complex, some with a nucleus, some without, but all of them following the same fundamental rule laid down over a century ago. Every cell comes from a cell that came before it.